Have you ever wondered how chickens can be so variable? Well, that's down to an amazing and beautiful process called meiosis, which is the genetically important part of making eggs and sperms. How much potential variation can meiosis cause, do you think? 1,000 genotypes? A million? A billion, maybe? No, not close. Keep going. There are over 549 billion possible combinations of maternal and paternal chromosomes that can be created via meiosis of the chickens. And then there's a special process called crossing over that creates yet more variation, and that's before you even think about allele diversity. If we could have $1 for every possible combination, we'd be much, much richer than Elon Musk was this year. Better not mention that to the chickens or they might start getting ideas. So, join me today for a quick look at the essentials of meiosis and learn how this wondrous amount of diversity happens. Understanding meiosis just a bit helps us to understand inheritance a lot more. Plus, later on, I will reveal a tip about egg incubation from a scientific publication I read while making this video. This video, it should be said, builds on previous videos of the series, especially videos 2 to 5. So you might want to review those first, especially if you find anything a little difficult. Otherwise, I hope to see you at the other end of this video. We'll look at meiosis just enough to understand how inheritance occurs in chicken. Meiosis causes the division of a body cell into 2 and then 4 gametic cells, which is the general term for eggs and sperm and their precursor cells. Because meiosis can seem complicated enough, we're going to consider a simplified version in a cell where there are just 2 pairs of chromosomes. The paternal chromosomes are shown in blue, and the maternal ones are shown in purple. This is the beginning of meiosis. The nuclear membrane has dissolved and the chromosomes are starting to condense and no longer look like a ball of string. This is a little later when the chromosomes have replicated and the maternal and paternal homologues have paired up in the central plane of the cell. When the paternal and maternal chromosomes of each pair come together, they may exchange parts with one another, which is a process called crossing over. This occurs in such a precise way that the recombined chromosomes neither lose or gain information, but simply carry new combinations of different alleles that weren't present in either parent. That's to say that alleles are swapped, so the maternal chromosomes will carry some alleles originally present in the paternal chromosomes and vice versa. This creates new combinations not present in the body cells of the parent. It shuffles the deck of DNA, giving extra variation in the chicks compared to their parents. By processes we don't have to worry about, the recombined chromosomes are pulled to opposite sides of the cell. The cells divide into 2, with each new cell having 1 chromosome of each homologous pair. Where there was 1 diploid cell, there are now 2 haploid cells. You should notice, though, that each chromosome is still double in structure. It is during the second part of meiosis that these cells are divided again, with each cell inheriting 1 part of the double structure of the chromosome. Each original diploid cell has now been split into 4 haploid daughter cells, each with a unique gene combination. And now for the amazing bit. If we ignore crossing over for now, the cell in our example could form 4 combinations of maternal and paternal chromosomes in its gametes. The number of possible combinations increases with the number of chromosome pairs. For chickens with 39 pairs of chromosomes, there are more than 549 billion possible combinations of maternal and paternal chromosomes. This is approximately 20 times more than the number of chickens in the world, and it is not including the extra diversity from crossing over and from genes often having more than one allele. Now, our birds have mated while we were messing around with numbers, and we'll return to the genetics of their reproduction. From previous videos, I hope you remember that the body cells of a chicken are diploid, with 2 sets of chromosomes. Thanks to meiosis, eggs and sperm are haploid, denoted by N. The union of the egg and the sperm forms a zygote and restores diploidy. After about 5 hours, the zygote starts to divide. This division starts before the egg is even laid and continues as the egg travels down the oviduct. Once the egg is laid, it cools down and division stops, resuming only if the egg is placed under favourable conditions, either in your incubator or under the chicken as she starts to sit. While the egg is cool, it's in a phase called embryonic diapause, where development is arrested. In the wild, diapause occurs in freshly laid eggs until the last egg of the clutch has been laid. This gives an evolutionary advantage to hens that can synchronously hatch their eggs. We use this phenomena as well to collect and store eggs before we put them in an incubator. Here are some things I hope you will remember from this video. A chicken's body cells are diploid with 39 pairs of chromosomes. The eggs and sperms and their precursor cells are called gametic cells and have just 39 chromosomes. The process of meiosis creates gametic cells out of body cells, mixing up maternal and paternal chromosomes to create variation. Crossing over during meiosis creates extra variation by creating chromosomes that aren't quite the same as either of the parental ones. The fusion of an egg and sperm during fertilization leads to a diploid zygote, which could be the beginning of a new and unique chick. The alternation of meiosis and fertilization in the life cycle maintains the constancy of the chromosome complement from one generation to the next one. Now we return to the tip I found while looking up something on diapause. Like many people, I store my eggs upside down while I'm waiting for enough of the right eggs to incubate. I try not to store them for more than a few days, being aware that the chances of hatching does decline over time. However, the research found that eggs stored at lower temperatures of about 12 degrees lasted for much longer than eggs stored at about 18 degrees, which is about room temperature. The eggs stored at the lower temperature could resume normal development even after 28 days. The take-home message is that if you need to store your eggs prior to incubation, keep them in a cool place. Thank you very much for watching this video. If you have found it useful, please like, comment below, and share it with your friends or on social media. Subscribe so that you're notified of Chicken Genetics videos as they come out. I hope to see you very soon.